Beneath the waves, a variety of underwater ecosystems silently perform functions that are essential to the health of our planet. Seagrass meadows are one of the most productive and crucial environments on Earth. Found on every continent except Antarctica, these marine angiosperms act as the backbone of coastal ecosystems. Despite covering less than 0.2% of the ocean floor, seagrass meadows store up to 10% of the ocean's carbon, playing a vital role in mitigating climate change. They improve water quality, protect shorelines, provide habitats for marine life, and support coastal fisheries and communities. However, these ecosystems face numerous threats, including seagrass wasting disease. Seagrass wasting disease is caused by a single-celled organism called Labyrinthella zosterae, which infects seagrass blades and hinders their ability to photosynthesize. In the 1930s, a devastating outbreak led to one of the most significant North Atlantic seagrass declines in recorded history. Despite growing information and monitoring of seagrass wasting disease, it remains a major threat to seagrass health. In recent years, rising ocean temperatures have created the perfect conditions for the spread of the disease, leading to large-scale die-offs of meadows. Massachusetts is home to 9,477 hectares of Zostera marina, the dominant seagrass species. This region is a critical case study area due to its temperate climate and the ecological, social, and economical importance of its eelgrass meadows. Under the Boston University Coastal Ecosystems class, we sought out to answer two main questions. How does seagrass wasting disease affect seagrass meadow structure and plant health in Massachusetts? And how does the disease impact the ability of these meadows to sequester carbon? We hypothesized that meadows with greater severity of wasting disease would show weaker structure in the form of shorter canopy heights, sparser coverage, and lower shoot density, and as a result, lower carbon sequestration rates.
All of the plant samples that we got for tonight and then analyze them.
Our study was conducted across 4 sites in Massachusetts.
It's right here! Should we start it? I think we should start in between both.
Pleasant Bay in Cape Cod, Cambridge Beach in Cape Ann, and Monomoy and Matticat Beach in Nantucket Island. At each site, we set up a 25-meter transect parallel to shore. Every 2 meters, we laid down a 0.5-meter quadrat and placed a smaller 0.25-meter quadrat within it. We carefully ran our hands through the bed and visually estimated how much of each quadrat was covered by seagrass. We took down measurements such as the presence or absence of algae and what percentage of the seagrass was covered in epiphytes, which are organisms that grow on seagrass blades. We tallied healthy and unhealthy shoots within our smaller quadrat to determine shoot density and the percentage of seagrass infected with SWD. We extracted 5 healthy and 5 unhealthy seagrass plants from each site. At the exact point we extracted our plant samples, we inserted a 10-centimeter sediment core into the These cores were split by depth for every 2 centimeters. We aimed to analyze how the carbon storage in the sediment varied depending on whether a healthy or diseased plant was growing there.
One time we're like,
I'm trying to make it so that
I thought you were going to start your intro.
people are.
Oh, the intro? No, it's...
Like, we're kind of in between.
Okay, she's literally a block of ice.
Back at the lab, we shifted our focus to analyzing plant characteristics and the sediment cores for carbon. For each plant, we measured sheath lengths, and leaf widths. We used the wasting index key to assess the severity of SWD on each leaf. In order to determine the amount of carbon sequestered by each plant, we followed various steps. Firstly, we dried the sediment cores in an oven for at least 24 hours. Weighing the bags allowed us to derive bulk density, which is the mass of sediment in a given volume. After weighing, we used a grain splitter to separate each sample into 2 bags for randomization. Next, we had to prepare our samples for loss on ignition, or LOI, testing. We ground and weighed approximately 6 grams of each sample before they entered the furnace. We burned the samples at 400 degrees Celsius for 16 hours. Afterwards, we weighed them again to find out how many grams of mass was lost. This mass loss is the amount of organic carbon stored in the sediment samples through seagrass photosynthesis. By taking the mass loss divided by the weight of the sample before it entered the furnace, multiplied by 100, we were able to find the percentage of organic matter in each sample. We used the percent organic matter as a proxy for percent carbon sequestered in our analyses. Next, we put our sediment in a mechanical shaker that passes the grains through a stack of sieves, sorting the samples by phi size, running from -3 to 4. We weighed the individual amounts of sediment that were now separated by grain size. This allowed us to find out what type of sediment the seagrasses were growing in.
Pleasant Bay is statistically different from Monomoy and Cambridge. And since Matticat has an AB, that means that it is so.
Our last step was to analyze our data using RStudio. Ready?
Yeah.
Okay, so now
Science.
we conducted an analysis of variance, or ANOVA, on our plant morphology and transect data, followed by a post hoc test to compare group differences. Additionally, we performed different regression analysis between the variables relating to seagrass wasting disease. Here is what we found. Firstly, we found a difference in wasting disease percent cover across the beds in Massachusetts. Pleasant Bay had significantly less wasting disease cover than the other sites, at around 25%. Cambridge Beach, Monomoy, and Matticat all had around 75% wasting disease cover in the meadow. The extracted plant samples in the lab confirmed that Pleasant Bay contained less wasting disease. However, Matticat showed less severe wasting disease on the actual seagrass blades. Overall, with the data from all 4 sites combined, we observed that wasting disease is related to the surface area of the plants. A larger plant will have a higher wasting disease index. When we break it down, this relationship is driven by the data found at Cambridge Beach. Looking at the various meadow characteristics, we saw no relationship between epiphyte cover and wasting disease cover. This is consistent across all sites. There was also no relationship between shoot density and wasting disease cover. This was seen at every site as well. In addition, we found a negative relationship between seagrass cover and wasting disease cover from the combined data of all sites. The lower the seagrass percent cover, the higher the wasting disease percent cover. This means that the more seagrass present indicates a healthier bed. However, this was not consistent at the site level, which may indicate that more data needs to be collected. Looking at the sediment data, we found that overall, as bulk density decreases, wasting disease percent increases. However, this was not seen at the site level. A higher bulk density indicates more compact sediment, which is usually a limiting growth factor because it reduces pore space, root oxygenation, and water infiltration. In addition, through the grain size analysis we conducted, we found that the majority of the sediment in Madaket Beach is a fee size of 1, which is medium sand. Lastly, from sediment cores taken between 2 and 4 centimeters at depth, we saw no relationship between percent carbon storage and percent wasting disease across all sites. The data we gathered shows that wasting disease is tied to a couple of plant characteristics, but actually has no effect on carbon. It is notable that Cambridge Beach showed several differences to other sites, which might be due to geographical differences, given that Cambridge Beach is located further north. For this site specifically, it is likely that more data points would lead to a significant result, showing that greater wasting disease relates to a lower percentage of organic matter.
In conclusion, our study demonstrates that seagrass wasting disease significantly impacts seagrass meadows across Massachusetts, affecting plant health and meadow structure. However, the disease does not significantly impact carbon sequestration, with its effects on carbon storage remaining inconclusive. These findings highlight how much remains unknown about how seagrass wasting disease affects these vital ecosystems. More data, especially with a timescale, would provide greater reliability and insight to scientific findings. It is clear that continued studies are essential to understanding the complex dynamics at play. Further research, greater public awareness, and widespread conservation efforts are crucial to the protection of seagrass meadows. By safeguarding these coastal ecosystems, we can move forward in our fight against climate change.
Which one do you like, the seagrass?
Don't let seagrass become seagone.
So bad.
I feel that as non-STEM majors, we did a great job.
Period.